Welcome to Biochemistry of Metabolic Processes. Today, we are going to explore a fascinating and essential survival mechanism: how our bodies adapt during periods of fasting and starvation. Have you ever wondered how you can sleep for 8 hours or even fast for several days without your brain shutting down from a lack of energy? Let us dive into the biochemistry and find out exactly how this works. To understand this process, imagine your body as a car on a long highway. When the main fuel tank, the food you just ate, runs empty, a built-in reserve tank automatically kicks in to keep the engine running. In the human body, this early fasting state begins roughly 4 to 18 hours after your last meal. As blood glucose and insulin levels drop, a hormone called glucagon rises. This hormone acts as a crucial chemical messenger to unlock your body's energy reserves and keep you conscious. Now that the glucagon alarm is ringing, the liver gets to work using two main biochemical pathways to maintain stability. First is glycogenolysis, where the liver breaks down its stored glycogen into free glucose molecules. But these stores are quite limited. As they dwindle, the liver also ramps up gluconeogenesis, which is the synthesis of brand new glucose from non-carbohydrate sources, like lactate and amino acids. Together, these two pathways ensure a steady, uninterrupted supply of glucose to tissues that absolutely need it, especially the brain. You might be wondering why this specific metabolic shift is so critical for our daily survival. Without the ability to mobilize stored glycogen and synthesize new glucose, our blood sugar would plummet dangerously low between meals or while we sleep. This leads to a severe medical condition called hypoglycemia. Severe hypoglycemia can cause confusion, seizures, coma, and eventually death. This happens because the brain simply cannot function without a continuous, reliable supply of energy derived from glucose. Since our liver glycogen is a limited reserve, we can mathematically estimate exactly how long it will last during a fast. The time until depletion, represented by T, equals the total mass of stored glycogen, M, divided by the rate of glucose consumption, R. Conceptually, this formula tells us that the duration of our reserves is directly proportional to how much fuel we have stored in the liver and inversely proportional to how fast our brain and organs are burning it. If your consumption rate increases due to exercise, the time you can fast decreases rapidly. Let us see this formula in action with a concrete example. Imagine a healthy adult male who just started fasting. We are given that his liver contains 90 grams of stored glycogen, and his body consumes glucose at a steady rate of 5 grams per hour. We need to find out how long his stores will last. We substitute 90 for the mass and 5 for the rate. Dividing 90 by 5 gives us 18. This means his liver glycogen will be completely exhausted in exactly 18 hours, explaining why gluconeogenesis must start well before this limit is reached. But what happens when fasting extends beyond a couple of days and officially turns into prolonged starvation? At this critical point, the liver's glycogen reserves are completely gone. While the body has massive energy reserves stored in the form of fat, there's a major biological problem. Large fat molecules cannot cross the protective blood-brain barrier. The brain is starving and desperately needs energy, but it cannot use the abundant fat directly. The body must find a workaround to survive. To solve this dangerous dilemma, the liver acts as a biological refinery. It takes acetyl-CoA, derived from the breakdown of fatty acids, and channels it into a specialized pathway called ketogenesis. This pathway produces water-soluble molecules called ketone bodies, specifically acetoacetate, beta-hydroxybutyrate, and acetone. Because they are water-soluble, these ketone bodies easily cross the blood-brain barrier and enter the brain cells. Once inside, they are converted back into usable energy to keep the brain functioning smoothly without relying on glucose. This production of ketone bodies is arguably our most important survival mechanism during prolonged starvation. Why is it so vital? Because if the brain continued to rely solely on glucose during a long fast, the body would be forced to tear down its own skeletal and heart muscle at a massive rate just to provide amino acids for making that glucose. By switching the brain's fuel preference to ketones, the body spares its functional proteins, allowing humans to survive for weeks or even months without food. We can actually quantify this incredible protein-sparing effect using a simple mathematical relationship. The mass of muscle protein saved per day, P saved, is calculated by taking the energy the brain derives from ketones, dividing it by the energy yield of glucose, and multiplying by a protein conversion factor. In plain English, we are first calculating how much glucose the ketones are replacing and then multiplying that amount by the protein cost required to make that glucose. The more ketones the brain uses, the more muscle tissue is saved, dramatically extending survival time. Let us calculate exactly how much muscle is saved during the third week of starvation. We're given that the brain uses 400 kilocalories per day from ketones. The energy yield of glucose is 4 kilocalories per gram, and it takes 1.75 grams of muscle to make 1 gram of glucose. First, we divide 400 by 4, meaning 100 grams of glucose are no longer needed. We then multiply this by 1.75. The result is 175 grams of muscle protein saved every single day, which can be the difference between life and death. Now, let us visualize this entire metabolic journey. On this graph, the horizontal axis shows the days of starvation, and the vertical axis shows the concentration of fuels in the blood. Notice how blood glucose, the blue line, drops slightly but then stabilizes. Free fatty acids, the red dashed line, rise slightly. But look at the green line, representing ketone bodies. It skyrockets after the first few days and plateaus around day 20. This dramatic rise perfectly illustrates the body shifting to a fat-based economy to keep the brain fueled. Let us summarize what we have learned. In the early stages of fasting, the liver acts as a generous provider, breaking down glycogen and synthesizing new glucose to keep the brain functioning. As fasting turns into prolonged starvation, the body brilliantly shifts to a fat-based economy, producing ketone bodies. This critical switch provides an alternative fuel for the brain, drastically reducing the need to destroy vital muscle tissue. You now understand the incredible biochemistry that keeps us alive when food is scarce.